The origin of the mitochondria and chloroplast can be explained by the endosymbiotic theory.
The mitochondria and the chloroplast organelles existed as their own prokaryotic cells. The two dodgeballs represent the prokaryotic cells. The endosymbiotic theory states that a eukaryotic cell engulfed an aerobic prokaryote, developing into a mitochondria, and engulfed a photosynthetic prokaryote, which evolved into a chloroplast. Evidence that supports the endosymbiotic theory is that mitochondria and chloroplasts have a similar shape and similar size. The endosymbiotic theory is also supported by their possession of a double membrane and the presence of DNA in both organelles.
Cells have often been related to cities, with the mitochondria being the powerhouse of that city.
The mitochondria is composed of an inner and outer cell membrane. The outer membrane is a simple phospholipid bilayer containing protein structures called porins. The inner membrane is where various chemical reactions take place. It is freely permeable to oxygen, carbon dioxide, and water. The folds of the inner membrane are organized into layers called cristae. The cristae are important because they increase the total surface area, enhancing the productivity of cellular respiration.
The membranes create two compartments, the inner membrane space and the mitochondrial matrix. The primary function of the inner membrane space is oxidative phosphorylation. The matrix is the fluid inside the mitochondria which contains enzymes responsible for the citric acid cycle. The matrix also contains mitochondrial ribosomes, tRNAs, and the mitochondrial DNA. Mitochondrial DNA is circular, covalently closed, and double-stranded. It is maternally inherited and it helps mitochondria divide independently from the cell.
Chloroplasts are composed of two membranes. The outer membrane is semiporous. It is only permeable to small molecules and ions which diffuse easily. The inner membrane regulates the passage of materials in and out of the chloroplast. Here, fatty acids, lipids, and carotenoids are synthesized. The inner and outer membranes form a border to the stroma. The stroma is an alkaline aqueous fluid where the Calvin cycle takes place.
Suspended in the stroma is the thylakoid system, which is a system of membranous sacs called thylakoids. Thylakoids are arranged in stacks called grana, where chlorophyll is located. They are the site for the process of light reactions in photosynthesis. There are two types of thylakoids: stromal thylakoids, which are the form of helicoid sheets, and granule thylakoids, which contain the photosystem II protein complex, which allows them to stack tightly and form many granule layers. This increases the stability and surface area for the capture of light. In the experiment conducted by Cheng, Sol, and Boutler on chloroplast gateways, it was supported that phosphorylation in chloroplast is regulated by different protein complexes. This evidence supports the wide array of cellular processes.
According to scientists Li, Kim, and Huang, mitochondria and chloroplast have membrane proteins that are responsible for transporting metabolites, ions, and other proteins to different parts of the cell.